of fibrous proteins. Those are common, but not, we've got a lot of collagen, but the interesting stuff is happening inside the cells, and you don't find fibrous proteins in there. Typically, they're all globular. These are smaller, not long ropes of molecules. They're wound up in little balls, typically water-soluble. And these are made out of alpha helices, turns, and beta sheets, held together by hydrophobic interactions of R groups in the center. So here's a small globular protein found inside the cell, kind of typical of a small protein. We've used that cartoon approach to show the secondary structure. So here's a bit of alpha helix, random coil, and this looks like the N-terminus. So a little alpha helix, random coil. Then we go into beta strand. It's anti-parallel beta strand that turns and does a U-turn, goes up there. The third beta strand, and then it finishes with an alpha helix. Common way of making globular proteins. The stuff here in the center would have hydrophobic R groups. The stuff on the outside would have polar R groups that love to interact with water. So the yellow is hydrophobic, and when the polypeptide chain folds, it folds in such a way that everything in the center is nonpolar. Sometimes you see some nonpolar on the outside too, but the center is almost exclusively nonpolar R groups, the so-called hydrophobic core. And so all of our cellular proteins inside the cell are built this way. They'll have at least two layers of secondary structure elements that come together with hydrophobic stuff in the middle. Kind of like a peanut butter sandwich, sticky peanut butter holding together layers of bread. So sometimes it's a two-layer sandwich, just a couple bits of secondary structure glued together with hydrophobic stuff in between. Sometimes 3 layers, sometimes very complicated multi-layer proteins with hydrophobic stuff in between the layers. Stuff on the outside is mostly polar, although not exclusively. Sometimes you see hydrophobic groups scattered on the outside too, but a lot of polar on the outside. The rule you can say with certainty is the stuff in the center, the R groups are never polar, they're always hydrophobic in the center. People categorize them, there are catalogs of these things. Here blue is the alpha helix, so the alpha helix proteins might have mostly alpha helix, but a little of something else thrown in there. Beta sheets, parallel guys often form barrel structures. That is a very common motif for enzymes. This top part of the barrel will be an active site. It's not important that you know these different, identify them. You should be able to look at one of these and say, well, this is a parallel beta sheet mixed with alpha helix. That's basically all I want you to know.
So just identify it as a tertiary, it's not enough. Right?
Well, it's a globular protein.
Oh, sorry, as a globular is not enough.
Well, you should be able to say that this is an alpha helical protein.
Okay.
Right? What holds it together? The usual suspects. All quarter long we've been talking about these weak forces: hydrogen bonds, hydrophobic, electrostatic, van der Waals. Certainly hydrogen bonds hold the secondary structures together. But I underline this for a reason. People consider this the primary thing stabilizing the fold, the tertiary structure. It's held together by this tendency to bury hydrophobic groups. The actual shapes, whether or not this is a helix or not, that hydrogen bonds play a big role in that, but the hydrophobic effect is the principal driver of tertiary structure. The side chain groups on the surface, they hydrogen bond with water. Everything in the center, all the backbone elements, all the carbonyls, and the amide hydrogens, in the center of the protein, you've got these two. Even if the R groups are hydrophobic, these guys are polar, and they're in the center of the protein. So the rule is these guys have to be hydrogen bonded to each other in beta structures or at alpha helix so that you mask the polarity in the center. Again, the side chains in the interior are almost exclusively hydrophobic. I think we'll skip that. If you're interested, when we talk about enzymes, you'll see proteins have to be non-static. They have to move. This guy at Yale has a nice website where he's made movies of proteins moving around. It's kind of cool. Last thing here is quaternary structure. So this tertiary structure is the folding pattern. What's quaternary structure? What's left? Well, some proteins have quaternary structure, others don't. Quaternary structure refers to multiple chains. A classic example is hemoglobin. It's got 4 polypeptide chains. So you have to ask the question, how do they pack together? There's different ways of doing that. They could be in a blob, they could be in a flat plane. The description of how those separate subunits pack is quaternary structure. Just tells you the number of subunits and how they pack. Okay, so this slide on hemoglobin kind of sums it up. All these different layers of structure. The primary structure is the sequence of the amino acids in the chain. Hemoglobin has a lot of alpha helical structure. That's its secondary structure, these alpha helices. The tertiary structure for the hemoglobin protein is the exact way in which these elements of coils pack against each other. How long are the coils? What angles do they make? The final overall shape, that's tertiary structure. But hemoglobin doesn't act as just a single protein molecule. 4 related ones come together to make a tetramer. 4 different molecules, each with their own amino terminus, come together in a special way. That's quaternary structure. So in the world of cellular proteins, many of them are monomers, so they have no quaternary structure. Some are dimers, a smaller number are tetramers. They tend to be even numbered for some reason. You see a few trimers, but so quaternary structure doesn't apply to everything, only to certain proteins. And at the end here, I threw some stuff in on gluten, figuring you guys would be interested in it. Gluten is an aggregation of different proteins. Glutenin and gliadin. They're in seeds as separate molecules. When you grind the seed open and expose it to water, so they like flour and collect the flour, these guys start interacting to make gluten. The glutenin is long and big. The gliadin is smaller, more water-soluble. In the presence of water, if you start kneading dough, they get linked together. They start forming disulfide bonds between them. Kneading the dough exposes air to all the insides. More oxygen means more disulfides forming.
They actually say that you can rinse the dough